Good morning, ladies and gentlemen. My name is Helena Kandarova. I am from the Institute of Experimental Pharmacology and Toxicology of the Slovak Academy of Science. I would like to thank organizers for the invitation to deliver talk on the validation of new in vitro alternatives to animal tests. And we will be talking about approaches to the validation, technologies that we have for the validation, that is available for the validation, and we will also spend some time on discussion on challenges in validations, and I will try to provide some hopefully positive outlook. There are several technologies that scientists try to validate for various purposes. The very simple biological systems start with the primary or permanent 2D cultures. Then more advanced systems are co-culture systems and spheroid. And I would say that the most advanced systems that are currently available for researchers and that are also entering validation studies are reconstructed 3D tissue models, chips, and microfluidic systems. We, of course, also have ex vivo models and methods that are based on ex vivo models, and these include isolated animal or human tissues and organs. From the non-biological test systems, there are several methods that underwent already validation studies, like the ROS assay, reactive oxidative species assay, DPRA for skin sensitization. There are also a variety of other analytical methods that underwent different stages of the validation process. And of course, in the 21st century especially, we are challenged by the computer simulation and mathematical models. There are QSARs systems, machine learning systems, and systems that are based on the artificial intelligence. So they also require a special type of the validation approaches. There has been also great progress, especially in the past 10 to 15 years, in developments of the different microfluidical systems and human on the chip concepts. These are fantastic technologies that will help us to develop more advanced in vitro systems that could be used in the systemic toxicity testing or in some very specific fields. However, with this increasing level of the technology, there is also increasing level um, on the complexity of the validation concepts that are in place. And most probably we will have to rethink the way how we run validations, because these systems will require a slightly different concept. But I will be talking about this a little bit later when we will be discussing the challenges. Great advances have been also achieved in the way how the models are manufactured. And the additive manufacturing, or also called 3D printing, is bringing a lot of new opportunities. The 3D printing can improve the quality of the models. It can accelerate the production process. It can also help us to standardize the tissue models. There are also, however, some new challenges that are coming into the production, and this is the biopolymer technology, because the cells during the 3D printing are embedded in the biopolymers. So there can be some influence of these components on the final quality of the tissue models. And also the printing machines is something that we need to consider as a component of the test model development, and also a component that may influence the quality and the reproducibility of the model itself. So let's talk about the validation of these technologies. This is a nice picture that I found in the internet that explains some of the components of the validation process. So validation needs to have a purpose. It also needs to have research. It requires certain procedures and development, and it should definitely result in a quality. So if we achieve all these 5 points, we are on a very good way to the successful validation. We need to understand first what we mean when we talk about the validations, and there are several existing definitions that are published in the literature. For me, one of the best definitions is given here. It's coming from the OECD document on the validation and international acceptance of new or updated test methods for hazard assessment. So it says that the validation is a process that ensures that alternative methods developed by academic or industrial scientists will be scientifically valid and thus eventually accepted by regulatory authorities for classification, labeling, product approval, or safety testing purposes. Validation is basically a process where we build confidence in our research and acceptance of our research. And validated methods are required to generate toxicology data in many fields, including the transport regulations, occupational safety, classification and labeling purposes, like in European Union, the REACH legislation, cosmetic directive, pesticides regulations, pharmaceutical guidelines on preclinical testing of the drugs, ISO standards for medical devices, food and feed regulations, and there are still many other areas where I didn't mention, but they also do require certain methods and certain test systems to be validated to be able to accept data that they generate. We academics usually see the validation as a kind of side event that makes the scientific route interesting, sometimes also very challenging, and which sometimes leads to the improvement of the methods or models. And I work in the area of the skin models, tissue engineering models, and here I can tell you that the competition for the best skin model prize is running already for more than 30 years. And it really led to the development of the better models with better barrier properties, models that are better resembling the nature of the skin. So competition is not bad, even in the academic area. Validation can be run by different parties. It can be validation centers, for instance, in European Union, it could be the ECVAM. Very frequently, validation studies are run by industry, and those are most frequently the commercial technology developers that have certain models, certain methods that they would like to introduce on the market. But also academics, they also run validation studies. Usually these are quite enthusiastic researchers that have uh, time and endurance because the validation may take several years, as you can see on this picture. Validation types are basically two. It can be prospective study, where you go from the research through the method development, pre-validation, validation, review to the regulatory acceptance. Or it can be also retrospective. That means you basically summarize data and look retrospectively whether the method is providing the required level of the reliability and relevance. In a validation study, it is not really prescribed that you have to use the GLP, good laboratory practice. But I would strongly recommend to at least adhere to the principles of the GLP or GIVIMP. It is of a great advantage, especially in case that you are optimizing the method. You need to really document every week what you have done, what you changed in your protocol or in your test system. But it also significantly helps you in the later stages when you go through the process of the review and the regulatory acceptance process, because in this stage you may be asked to provide all the raw data in addition to the report that you are submitting to the regulators or to the validation party that evaluates your test method or test system. As I said, validation may take quite long. From personal experience, it may take from 9 to 25 years. I have been working in several validation studies, in some of them as a scientist, and in other studies as a manager. And I can tell you that it's not really usual that the validation process is running straightforward. Many times you have to step down from the validation to optimization of the test method, and sometimes also the review and the regulatory acceptance takes a bit longer. So person that is willing to work in this area and would like to validate the method that has been developed in the laboratory needs to be prepared that this is a very long process and that may take many years until the method will be regulatory accepted. In 2004, Thomas Hartung and his team from Euro-Equam published a very good paper on the modular approach validation. It is approach that tries to simplify a bit the validation process and break it down to the modules that can be performed in parallel. So there is no more requirement for sequential assessment. Although I still think that to make the validation effective, some sequences should be maintained. So he and his team broke down the validation study into 7 modules in which the reliability and relevance are addressed. It is run by the validation team management, and if all these 7 modules are fulfilled, then it goes to the independent peer review. I would like to spend some time on some of these modules because fulfillment of these modules is the requirement for the success of the validation study. The first thing that someone who is developing alternative method needs to take into the account is the test definition. One has to be really sure what is developed in his or her lab. The test needs to be well described. It needs to have an SOP or protocol according to which the test is conducted. The test system needs to be really well characterized and it needs to be reliable and relevant to um, the endpoint that is going to be addressed. One has to choose also correctly the endpoints and biomarkers that will be measured within this test methods. And the choice of the endpoints and biomarkers can be very important to correctly describe the relevance of the method. And the prediction model, that is the third part, that is a key component of the test method. If the method doesn't have a prediction model, then it is not really yet ready for the validation. The prediction model needs to be part of the protocol, part of the SOP. It needs to be developed on a basis of some data set. Usually you have a training set and then you have a testing set. This testing and training set should be chemicals that are well representing the broad spectra of the chemical universe. So it's very, very important to spend necessary time to develop a good test method with all these components. This includes also development of the negative control, positive control, reference items, and also development of the acceptance criteria. Without this, no method is ready to enter into the next step. What all can influence the test system reproducibility? So there are many things that can cause some variability in your test system. And you need to be very sure that you have all these components under your control. It could be the non-cellular components, it could be the basement membrane, the collagen. If you are creating skin models, it is the epidermis, dermis, for instance, some artificial scaffolds that you have in the system. Then the cells, of course, whether you have keratinocytes, fibroblasts, liver cells, or whatever cells, you need to very precisely control the parameters of your cells to be sure that they are not increasing the variability of the test method. Then the production process. If you have manual production process, there can be variability that is brought by the lab technician. If you have automated process, it's better. But also in the automated process, there can be some slight variations that can influence the final product. And of course, the experimental conditions and the endpoints measured. If you have static conditions, usually the methods are a bit more reproducible than if you have the flow inside, because the flow can also make changes in your test systems and may increase the variability if there is already variability introduced by the non-cellular or the cellular components. So one has to be very sure that these 4 things are well under the control when developing the test system. Then I also am frequently asked what is better to use, whether scientists should work on the commercial models or better homemade. So the commercial models, I would say, have certain advantages. They are coming into your laboratory with a certificate of the analysis, so you know that someone already checked a number of the problems that may be introduced into the laboratory and assures you that they are not present. And the manufacturer is also usually very closely manufacturing the quality and the reproducibility of the system. If not, then it is not a good partner for the collaboration in the validation project. So if you take models, cells, 3D tissue models from commercial source, make sure that they are always coming with the certificates of the analysis. Of course, it needs to be backed by the data, not only the statement, but you need to have access to the data, and that the developer is really keeping track on the positive and negative control testing of their system. If you have this commercial model, then you have something that is pretty well standardized in your laboratory. Of course, you are lacking the possibility to introduce something innovative into the test model systems, but you are sure that you are not developing a mermaid in your laboratory, right? If you are working on your home-made models, then you definitely know what is inside. You may use multiple donors that might be easily available. Especially if you work with some tissue culture banks or hospitals, you have more options and more choices to construct specific tissue that is suitable to address specific scientific questions. And usually, you have also more flexibility in planning on and conducting your studies. On the other hand, there is really high risk of non-donor related variability due to less stringent requirements for the biological material, cell culture media, and production components, and also non-adherence to the good manufacturing practice. That all may have a great impact on the variability of your test method. So be careful when you are using your own tissue models. Try to keep everything under the control and document everything. The second module, transferability, it's a module that needs to be addressed once a test method is really well described, well developed. It went through the initial testing in your own laboratory with at least 20, 30, 40 chemicals. Transferability basically is a check of readiness of your system to enter a pre-validation stage. In this process, in a transferability step, you are trying to bring your method and your SOP to the second laboratory. And the second laboratory should basically be able to produce the same data as you are producing in your laboratory. Part of this, and very important part that is many times underestimated, is training. If the training is not well conducted, there is really high risk that the transferability will fail, and later on that even the validation fail. Training should be usually two-phased. The laboratory that is introducing the test method to another laboratory should travel there, check whether everything is according to the requirements for the transfer, train the personnel that will be evaluating the method that is being transferred. And the second phase is basically test itself, where the laboratory that conducts the transfer is producing the data without the supervision. If the second laboratory produces the data with the same predictions as the test method developer, then this transferability step is fulfilled and people can progress to the next stage of the validation study. Again, I would like to stress that this step is very necessary to improve the clarity of the standard operating procedure, of the protocol, as it's called in United States, and it's really important step to see whether the method still needs some development. If the transfer went well, then we can start to discuss the next step, which is assessment of the within and between laboratory reproducibility. Within laboratory reproducibility is something that is always required. Basically, you need to run the test with different operators in different times. Usually 3 independent runs are performed, and depending on the complexity of your prediction model, the statistician calculates how many samples you need to run. Between laboratory reproducibility, this is the assessment of the reproducibility of the experimental data between 2 to 4 laboratories. It can be run on a part of the set of the materials or in the whole set of the materials. This depends on how much time and money the testing laboratories have and can invest into such a validation study. I have seen validation studies that usually had 3 to 4 laboratories, but I also participated in validation study where we had 21 laboratories, which was very interesting exercise. And also there in this large validation study, it was really pretty much visible which of the laboratories underwent training on site and which laboratories just tried to interpret the test based on the SOP. In assessment of the predictive capacity, laboratories basically need to reach the sensitivity and specificity towards an in vivo standard. In many cases, some sensitivity and specificity levels are already given by the statistician or by the regulators. They would like to see, of course, the highest sensitivity possible, but it also specificity should be high so that we do not have under-predictive or over-predictive methods. So sensitivity and specificity levels around 80 to 90 are usually accepted. Here we usually have problems with variability of in vivo data into which the in vitro systems are compared. Therefore, you really need to spend time on selecting materials that have good quality in vivo data, ideally from GLP studies, where you do have access to the raw data and explanations why there were certain deviations in some of the animals. It's really important to be able to check the effects that happened in the test animals, especially if there is discrepancy between the in vivo and in vitro result. The raw data, the raw animal data are of very special importance. And also, you need to count with some less than optimal correlation between the human and animal. Here I have a study for skin irritation testing where we dig deep into the data sets that were available for the rabbit skin irritation testing and human skin irritation testing. And we have seen that many of the materials that were classified as irritating in the rabbits were in fact non-irritating in the human volunteers. And when we developed a method for skin irritation testing with the reconstructed human skin tissue models, we have seen that we, in some cases, were much closer to the human response than to the rabbit response. So if you have possibility to get some human data from controlled exposures, that is the best data set that you can use to understand why your test method may not always match the in vivo data. So if possible, I would always recommend to discuss the clinicians, maybe some poison centers and so on, whether it is possible to get some human data in addition to what you have from in vivo testing in animals. I would say that the biggest success has been achieved in validation of alternative methods for the topical toxicity testing. There are a number of methods that successfully went through the validation process and are implemented in the regulations worldwide. Some people say that it is a long-hanging fruit, but definitely this is not the situation for those that worked over 20 and more years on these topics. I just would like to mention that first papers on the reconstructed human tissue models for the toxicology use are from early 90s, and it took 10 years to validate first in vitro methods based on the 3D tissue models. And even 20 years after the first publications, we are still not addressing all needs in the topical toxicity testing. So a lot of planting is still ahead. And I would not like to hear that this is the low-hanging fruit because it was a lot of work and many people benefit from what has been done by the people working on these validation studies. A lot of lessons were learned, and the work that has been done is now improving the concepts that are developed for the future validation studies. I have here, just for information, a table that is showing you the overview of use of the reconstructed human tissue models in hazard and safety assessment as of 2023. So you see that there are areas, especially for the skin corrosion, irritation, phototoxicity, and eye irritation, where we do have validated OECD methods. There are still other areas, like for the skin sensitization, skin penetration, and genotoxicity, where the in vitro technologies still need to go through validation. Some of them are in quite advanced stage of the validation studies, but it may take still 1 to 2 years until we see them in the regulation. So progress has been done. It has been a lot of work, and I do hope that we will see more validated methods, not only from the area of the topical toxicity, but also from the other areas in the regulations. There are many challenges, but also many opportunities that we have in the 21st century. And what has been a problem 20, 30 years ago, today is not seen anymore as a problem, but maybe as a new opportunity to develop something better, something advanced, maybe commercialize it. The first challenge that is remaining is that we still try in many cases to compare the in vitro data to the data that are produced in animals. And we do know that animals may not always represent what is happening in the human. I like to show this picture in my presentations, in addition to the one that I produced myself in my PhD studies. It's a picture that is showing the human and animal bioavailability, and it is basically a graph by James Harris, who presented it at the Center for Business Intelligence conference. I don't want to go into details, but you see how much the data are scattered, and for these drugs, there are not really um, I would say agreement, any agreement between what is happening in the human and between the animals. Of course, there are areas where we do see agreement, but we need to be prepared that in some areas the animal models really will not be the models of choice when trying to assess the sensitivity and the specificity. As I said at the beginning, if it is possible, we need to really look into the human data. The other two challenges or three challenges are linked to what we already know and don't know, especially for the systemic toxicity testing and for the development of the drugs. We don't know a lot about the microbiome that may activate or deactivate some medicines and some chemicals. So we need to spend a lot of time in acquiring additional knowledge on what is going on in our human bodies to understand the effects better and to be able to model the systems that may also include the microbiome in the consideration. Challenge three is the rigidity and relevance. So many regulatory bodies are usually not keen to accept changes, especially fast changes. That's normal, but we also need to think about what we are accepting and what is accepted. Our gold standards needs to be rethink because animal data are more and more challenged by the modern science and gaining knowledge. So if we have obsolete methods that are based on non-relevant animal data, we need to get rid of these methods from the regulations. And we need to start to validate against human relevant data with a human with exposure that is relevant for human. So that is something that will require a lot of discussions with the regulators, and I hope that the outcome will be positive. Because we cannot really achieve success if we will try to fit the modern assessment into the outdated regulatory frames. Challenge four is lack of finances, lack of communication, and lack of knowledge. As I said many times in my previous presentations, we are lacking finances to effectively run validation trials. So there needs to be something done with this. I hope that politicians will fund more validation studies through the, for instance, Horizon project. That would help quite a lot. But we also need to communicate better to the developers and regulators and also politicians about the need of the alternative methods. And we need to spend also time to educate our students and our colleagues how to effectively design and run validation studies, because this is also the piece of the success of the validations. If you know how to run it, you won the half of it. Challenge number 5 is the increasing complexity, in my opinion, because we are having specific complicated tools, and we don't have yet the validation concepts for them. So we need to rethink how we run validation studies and how to address the increasing complexity of these models and systems. And this is my last slide. So I think that depending on the objective of the study, the correctly selected alternative methods in combination with deep knowledge of the tested compound may be more appropriate methods for many areas of interest. So we can use this method instead of the animal test. Regulators and test methods developers are currently communicating much more effectively than it at any other time before. This, of course, is very positive change, and it leads to the accelerated acceptance of the alternatives, and we need to support this and nurture this communication. And for the future, I think that we do have exciting tools in our hands. They are very, very promising, and they will help us to overcome the gaps, especially for the organ and systemic toxicities. And as I said, we need to rethink the classical validation process and to get more trust to new technologies. I do hope that with this positive approach, we may be able to replace many, many in vivo methods by the in vitro test systems within the next 20 years. And with this, I would like to thank you for attention and maybe mention a quote by Charles Kettering, that there exist limitless opportunities in every industry, and when there is an open mind, there will be always a frontier. So please, test method developers, don't be afraid of the validations and go into it. Thank you.